Welcome, everybody. This is the second of the third talks, the third speakers today, and after this, pardon me, the third of the three speakers today, and after this, at 4 o'clock, there'll be a panel involving all three speakers, as in previous days. Steve Phelps is from the University of Texas at Austin. Everyone should know him already for his work on evolution and social behavior. And I'm not even sure what in particular in his huge oeuvre he's going to be covering in this talk, but I'm looking forward to hearing it. You've technically got 50 minutes and then questions.
Well, thank you very much, Steve, and thank you guys all for being here. I'm going to talk about work that's appropriate to the panel discussion, which is going to talk a bit about space use and social memory and attachment. My title is a little broader than that. I'm going to begin a little more broadly. Love in the Mind of Wanderers, What Prairie Voles Can Teach Us About Attachment. And this title, Love in the Mind of Wanderers, wanderers are a particular tactic that male prairie voles may take, and I'll talk about them in a moment. You may also recognize the title as a little bit of a play on the title of a famous novel by Gabriel Garcia Marquez called Love in the Time of Cholera. This is a really, I think, just a beautiful novel that I read, when I was in college, a few decades ago. The novel is about this couple that falls in love basically as children, as adolescents, and then are separated, and then it follows the life of these two, and the female character, Fermina, decides to marry another man, a man who is socially appropriate, and this man kind of represents the rational head, the sensible choice in life, and then her love for this other man, who's known as Florentino, represents something else. It represents something irrational and something deep and long-standing. And so the novel is told from Florentino's perspective, as he is shunned as she marries this other man. And then he spends his entire life sort of pining for her and longing for her. And in addition to the beauty of the writing, it captures really essential things about attachment, I think. The importance of memory, the sense of that emotional weight of it. And it really kind of brings to mind, I think, some questions about what attachment is, what love is, whether it's something that Garcia Marquez is actually sort of comparing it to disease, comparing it to cholera, and cholera is kind of a stand-in for passions more broadly. So I guess what I want to do maybe is begin this abstract, rambling introduction by asking you guys a question, which is a pretty simple one, I think, and hopefully many of you will be able to answer it. What's it like to be in love? And so I hope, in order to encourage you guys to stop and engage and ask questions, there's plenty of time for me to get through what I plan to get through. So feel free to stop at any time. But let's just start. How does it feel to be in love? Does anyone want to venture a description? Yeah. At the microphone, no less. Yes. What's that? Should I bring the microphone closer? Okay, I can do that. Thank you. It feels like an addiction. Can you describe that? What do you mean by that?
Well, for example, you'll see many cases where you can't actually get what you want, what you're hoping for, and yet you keep irrationally hoping that you might get it, even though you rationally know there's no way you could possibly get it. For example, that's one example. That's a little bit like craving for drugs. Although I've never been on drugs, I swear. I hear it's the same.
No judgment here, I promise.
But yeah, that's like, you ask one word, so
yeah.
addiction.
Okay. Yeah. Thank you. Yes.
One of my psychology colleagues described it as an abnormal pattern of behavior not yet entered into the Diagnostic and Statistical Manual of the Mental Disorders. And I thought, uh-oh.
Yeah, well, it's actually funny you should say that, because until the DSM-5, if I remember correctly, the depression that follows bereavement was considered something that um, that became an exception for the diagnostic criteria you would use to judge depression. Yeah, exactly. So think for a moment what it feels like to see somebody that you love, right? To see somebody, to return from a trip, for example, and to have your loved one greet you, or to see somebody that you're infatuated with or in love with in the same room. There's a spark of joy there, but there's also something else. There's something sometimes a little darker. There's a spark of longing, and if that longing isn't met, isn't reciprocated, then really it can be quite painful. As William James said, as long as one poor cockroach feels the pangs of unrequited love, this world is not a moral world. I think these aspects of love really speak to something really fundamental about its biology, and that I won't be by any means particularly original to suggest that love really does have something to do with addiction. And what it has to do with addiction is its dependence on reward. And so reward is something that, from a psychological perspective, simply serves to increase the probability of a behavior. And the reason we have rewards is because those behaviors that are increasing in response to reward are things that are generally adaptive. And so reward is a way to link environmental context to useful behaviors. And when we think about behaviors that are rewarding, they're things that by and large promote fitness: food, sex, comfort, positive social interaction. These are all things that are ultimately good for us. And these different kinds of rewards, despite the incredible distinctions that there are among them, generally rely on really common pathways. There are a handful of brain regions that we know about that are really important for reward and important for reward-related behavior. And in addition to these small number of brain regions, there are a relatively small number of neuromodulators that also govern reward. So we know about these brain regions from classic electrical self-stimulation studies, in which scientists put electrodes into the brains of rats, and they found places in the brain that animals would self-stimulate, and found that they would often do so in lieu of other sensible natural rewards. We know these compounds include things like dopamine. They include the opioid peptides in particular. They also include natural cannabinoids like anandamide, which have the function of seeming to kind of tweak the gain on natural pleasures. And so through the work of Kent Berridge and a variety of other folks, we have the sense that reward actually really has a couple varieties of it that, when we think about it closely, are a little like the distinction between between pleasure and desire. And so what they distinguish as the difference between liking and wanting. So liking is the actual feeling of pleasure, and there seem to be some really specific brain regions that are involved with the pleasure per se, and the opioids seem to have a special access to that. But then there's also the desire for pleasure, which we call wanting. And that sort of longing is this more aching aspect of pleasure. And that seems to be mediated heavily by dopamine. Well, if we think that we can translate this language into the language of love and attachment, then maybe we might see these same pathways being important in humans. And Helen Fisher, an anthropologist, really pioneered this when she did some imaging studies in which she asked undergraduates two simple questions. She showed undergraduates a picture of an opposite sex person, and that opposite sex person was either their romantic partner or some other individual. And then they asked them, how much are you in love with this person? And how attractive is this person? And what she found was that the answers to both of those questions were correlated with imaging activity measured by fMRI in this reward system, including the ventral tegmental area and the striatum. So it seems that some aspect of love and attachment really is about reward, and we know, and comparative psychologists have been studying reward for a very long time. Um, the idea that love is a kind of reward is a relatively more recent idea, at least at a neurobiological level, and it really has its origins in a peculiar animal model I know you guys have heard about earlier in the class. And that peculiar animal model is the prairie vole, and it's the prairie vole I'm going to be really focusing on today. So just to remind you that prairie vole is socially monogamous, which means that they form pair bonds and exhibit biparental care. Those pair bonds of biparental care don't necessarily translate into sexual exclusivity, just like in humans, right? Just like in lots of species. There's the process of bonding and attachment, but those are not synonymous with sexual exclusivity. Well, when they form pair bonds, that pairing is characterized by relatively small, actively defended home ranges. The males in particular really shrink the size of their home range and become very aggressive after they've formed a bond. We call these aggressive males residents once they've formed a pair bond, and we can distinguish them from the unpaired males we call wanderers. The wanderers maintain a much larger home range. They don't seem to use it in a way that's as intensive. They seem to literally be wandering, and they don't defend it against other individuals. Well, one interesting thing about the prairie voles is that even within a mating tactic, even within residents or within wanderers, there's a lot of variation in how individuals live in the world and how they use space and other aspects of their behavior. And so this variation in space use and sexual fidelity becomes kind of an interesting thing for us to explore and to think about individual differences in behavior and how it might relate to individual differences in the brain and how that might relate to individual differences in the genome or epigenome. So what we're going to do is we're going to really kind of drill down and we're going to focus on males, not because males are the whole part of the story, but because males are particularly important to the mechanism we're going to take as our focus. And that mechanism revolves around the hormone vasopressin. So vasopressin is a 9-amino acid peptide. It's closely related to oxytocin that you guys have heard about already, I'm sure, in Larry Young's work. It is implicated in a diverse array of species-specific behaviors, including pair bonding in the prairie voles, but also including aggression, social memory, aspects of courtship, vocalization, a variety of display-related things in many species. Well, the reason vasopressin seems to be important in terms of sex-specific behavior is that in many mammals, there's actually a sexually dimorphic population of vasopressin-containing neurons. And so if we look at a group of neurons that are in a structure called the extended amygdala, so they kind of go from the medial amygdala into the bed nucleus of the stria terminalis. This group of neurons expresses vasopressin in males, but not in females. And if you record from those neurons while you stimulate the accessory olfactory bulb, which receives pheromonal information, you can see that those neurons are getting direct input from those other neurons. And so the idea is that these vasopressin-containing neurons that are sexually dimorphic are listening to the presence of pheromones and then bathing the brain in vasopressin in response to that cue. And then what you do with that information, basically telling the brain that you're in a social circumstance. And what you do with that information depends on where in the brain you put receptors. And so if you put those receptors in reward regions, like the ventral pallidum, for example, then you change the sensitivity to rewards. And so the idea here is that if you look at prairie voles and you compare them to a promiscuous congener like the montane vole here, these are pseudocolor images that are showing the intensity of vasopressin receptors. And so in the ventral pallidum, you have lots of vasopressin receptors in the prairie vole and very few in the montane vole. And the idea is that that when they're with a mate, and they're mating extensively, they're smelling pheromones, they're listening vasopressin release, but then at the same time, they're getting the positive reward of the sexual behavior. And that by doing that repeatedly over the course of a day, they form a very strong reward association with the details of that mate, and that memory becomes a kind of bond. And now whenever they see or smell that mate, presumably they're evoking aspects of that memory and that emotional experience that's been facilitated by the presence of vasopressin. Well, that's a bit of speculation about exactly how it works, but what we do know is that if you put a vasopressin antagonist into the ventral pallidum, the prey voles mate perfectly normally. It doesn't interfere with that at all, but the mating doesn't translate into a bond. Similarly, if you take a virus and overexpress the vasopressin receptor in a normally promiscuous species, you can induce aspects of social preferences from mating that normally you couldn't do. And so together, the data suggests that this vasopressin receptor in the ventral pallidum is really causally related to the ability to form a bond. What's unfortunately, though, or maybe not unfortunately, interestingly, there are a lot of other places that differ between species. There are a lot of other places that express vasopressin receptors. So what I'm going to do is I'm going to talk about a really fascinating one that links not so much to reward, but more broadly to memory and to long-term memory. And this is a brain region called the retrosplenial cortex. So I'm showing it here. And so if you notice here, the retrosplenial cortex has quite a bit of expression of the vasopressin receptor in the prairie vole, but not in this promiscuous congener, the montane vole. Well, the retrosplenial cortex connects here to the hippocampus, a famous memory structure, as well as to the lateral dorsal thalamus and the anterior thalamus. It's part of a kind of memory circuit that is really important in a variety of memory tasks. Well, what's interesting about this is not just that there are species differences in the abundance of vasopressin receptor here, but when I went out into the wild and caught wild voles and looked at differences in their brains, one of the things that was really shocking was that even within a species, there were profound differences in the abundance of vasopressin receptors. So here on the right is a male from the brain from the upper quartile of natural expression. And now these aren't pseudocolor images. This is an actual autoradiogram. So what we do is we take the frozen sections and we incubate it with a radioactive hormone. And wherever there's lots of protein that recognizes that hormone, the radioactive hormone is going to stick. We wash off the extra, we dry it, expose it to X-ray film. And then wherever the film gets dark, there must have been a lot of radiation and therefore a lot of receptor. So what we have here is a brain that has a lot of receptor in this retrosplenial cortex. Also has it in the dorsal thalamus here, in the amygdala down here, in the hypothalamus. If we go over to this other brain, however, you see normal levels in the dorsal thalamus, normal levels elsewhere, but nothing at all in the retrosplenial cortex. Nothing at all. So the fact that it's here tells us that the gene for the vasopressin receptor works, it's intact, it's expressed in other places, but for some reason, the protein doesn't show up in this one brain region. So that became really interesting to us because this brain region had been implicated in spatial memory per se. It lights up during navigation tasks in humans, lots of other things. We have differences in the way animals use space. So we thought maybe it'll be related somehow to how the prairie voles use space and maybe these differences between residents and wanderers. And so we thought that this would be kind of a good way for thinking about a bigger question, a question that in traditional behavioral neuroscience we don't usually get to ask, which is a question about individual differences and what they mean and where they come from. If you think about the history of the field, you can think about a couple different folks that represent this history. On the left is Darwin. And in the Darwinian perspective, represented by, say, the classic ethologists like Tinbergen and Lorenz, in this perspective, the idea conceptually is to understand natural diversity. And with respect to behavior, is to understand the variation in behavior within and between species for its own sake, right? And that's really distinct from the aims of traditional psychology. So psychology, represented here by William James, is interested in using animal models, but for the most part, they're animal models. That is, they're there to help us understand fundamental aspects of psychology that then help us to understand the human psyche, right? And so that's a very different aim. It's not necessarily, the animals are really sort of proxies for us in that tradition. And as a result, and really it's been this comparative psychology tradition that by and large has been more successful at looking at mechanisms of behavior, at understanding the role of the brain and psychological processes. And so as a result, one of the strange positions we find ourselves in is that if you look at the distribution of research effort across the field, it's really unevenly distributed. So if you find, for example, that if you look at, this is something, a literature search I did a number of years ago, but I suspect it hasn't changed much. If you look for a variety of search terms related to social neuroscience and then categorize them into those, looking at human work, other non-human primates, rodents, et cetera, what you find is that about 40% of the work in social neuroscience would be with humans, and about 40% would be with rodents. But of course, that 40% with rodents is overwhelmingly lab mice and lab rats. So just a couple species, even though mammals themselves, or just the muroid rodents, are something like 40% of mammalian diversity. It's this enormously diverse group, which we really haven't explored at all. And so this uneven allocation of research effort puts us in a strange place, because even if, as psychologists, we want to use animals as models for humans, we often find that we don't really know how to generalize from one species to another, right? So if you're just looking at one or two species, you need some deeper phylogenetic framework to generalize from one species to another. And then on top of that, there are certain kinds of questions that are just fundamentally excluded from the approach. And so that if the species are genetically, if the species you're studying is genetically identical, or even if it's outbred but genetically impoverished, you can't really, it's not really good for asking questions about genetic variation. And of course, genetic variation is critical to all kinds of variation we see within any given species in nature. And so we're really kind of systematically excluded from asking certain really interesting biological questions. And just to give you a sense of what it might look like if an evolutionary biologist were to do neuroscience, keep in mind that if you have a trait, and so this could be any continuous trait. It could be some measure of memory. It could be some measure of speed. It could be height. You have some distribution of frequencies within a population. You have some kind of null expectation about what that might look like. And then if you subject that population to selection, maybe you favor one end of that, you see not only does the mean increase, but the variance shrinks, right? The heritable variation declines in response, at least in the short term, in response to selection. We know that's kind of a fundamental thing that happens. Other things can happen, like in response to different kinds of selection, the heritable variation might increase and the mean might not change at all. These are things that evolutionary biologists think about commonly, but it's really missing from the way we think about neuroscience and cognition and animal psychology, as well as much of human psychology. Well, for something like social cognition, though, this kind of change and variation could be really quite important. And if we look at the history of behavioral ecology, we can see some interesting examples. And so here we have a variety of species in which there are alternative male strategies, in which maybe a male has horns or doesn't. In the case of the beetles on the right, males may be sort of satellite males, as the case of the horseshoe crabs there. On the left, we have a fish that is in which some males are territorial and parental and others are not. An extreme example of individual differences in social behavior here come from these two tadpoles. And these are two tadpoles of the same species. This one, however, is about to eat this one. And that's because depending on the environmental details, the density of the population, the level of water, et cetera, they can switch between, they can choose either to be a conventional tadpole or can adopt this cannibalistic morphology. There are differences in the throat fans of lizards, associated differences in behavior, all kinds of really fascinating stuff. I'm going to tell you one last little example here, which is this lovely work that Michael Sheehan did. And so Michael has done a bunch of stuff on the recognition of faces in wasps, but as a postdoc, he did this clever thing where he looked at variation in the human face. And first he noted that the coefficients of variation for the human face were quite a bit higher.
Are you getting on the slides you want?
What's that?
Are you sitting on this one or?
Yeah,
Okay.
for a moment. Actually, no, this is probably the last, and I'm about to switch to another, but anyway, so he did this really lovely bit of work where he looked first at the coefficients of variation for facial features versus other parts of the body, and showed that the face seemed a lot more variable than you might predict based on simply the size of the nose or the ear or whatever. And then he looked at loci within the genome that had been detected as contributing to facial variation. And what he found was that the distribution of frequencies of those loci suggested that selection had actively maintained variation in the structure of the face. The idea is that the face gives you information about identity that's useful in signaling, and that selection's actually shaped that. It's actually kept that variation around. I think when we put all these kinds of things together, it suggests that maybe there might be variation in social behavior or social cognition that has a heritable component that selection might actually keep around, because the best thing to do, the best strategy to take within a social environment often depends on the distribution of other strategies within that environment. So it may be that this systematic overlooking of genetic variation could be really profound. It could have really profound consequences for how we think about the brain and mind. So we've got examples of alternative mating strategies I've mentioned across a variety of animals that are well known, but among humans, we've got differences in personality, differences in resilience. We've got the possibility of trying to tailor genomic, or trying to tailor interventions to the details of our genome. For a lot of reasons, thinking about genetic variation is really important. Another reason is the issue of neurodiversity. It's not exactly clear to us what even constitutes normal variation, because it's so far afield of the way we normally think about the brain and mind. And so if someone with an autism spectrum disorder or ADHD or a variety of other things, the boundaries between what we call disease and not disease is really a statement of values more so than it is necessarily a statement about what's normative. So the big question then that we're going to try to address with this work, using the retrosplenial cortex and the vasopressin receptor, is whether we can identify adaptive variation in the social brain. So here's a little overview of what's left. We're going to try and relate patterns of paternity and space use to differences in the brain. We're going to see if we can map brain phenotypes onto underlying genotypes. And then we're going to ask whether sources, we're going to ask whether sources of genetic variation have been actively maintained by selection. Any questions before I move on? Pause for a second. Stephen, did you have something that you needed to do here with the presentation? No? Okay. All right. Okay. I'm going to take you guys in the weeds here by the time we're done, so don't be afraid to raise your hand. So this is a prairie vole, and you see he's got a little thing around his neck, and basically that's a radio transmitter. And put that on a little zip tie that serves as a collar, and then we can go out and just measure where this guy is. He's inside a little aluminum trap there in this picture. And then this is Alex Ophir, who I believe you guys met a little earlier. Alex used to be a postdoc in my lab. And one of the things he did was to radio track these guys. And you just go out with an antenna, and you find where the signal is greatest, and you go somewhere else in the enclosure and do the same thing. You kind of triangulate and estimate the position of that individual. And if you're doing it manually, you can 2 or 3 times a day, you can get estimates of where individuals are for, 40 or 50 individuals. As you do that over the course of weeks, those individual estimates can be put together into a kind of probability density function that describes the likelihood that the animal will be at a given point in space, right? And then we can use those probability densities to estimate the likelihood that they'll encounter other individuals based on those probabilities. And so that's what we're showing here is one of those maps. And so the X and Y here reflect the dimensions of the enclosure. And then I'm showing probability densities for 5 different pair bonded males. And so the solid surface is the focal male. And then the wire surface are the territories of each of 4 other pair bonded animals. So this is one male, this is another male, this is a third, and this is a fourth. Oh, I see. Huh. Has my pointing not been showing the whole time? I just realized. Thank you. Well, that's okay. I can balance my speaking into the mic and my pointing. I'll go ahead and show you guys and yell a little bit, so it'll be not so great for the streaming. But oh, fantastic! Thank you very much. Okay, so this here in the solid surface is the focal male. These are the dimensions of the enclosure. And then this is one resident male. This is another resident male, a third, and a fourth. And then not shown here is the 6th male who's not pair bonded, but it's a wanderer. And he's not shown because he's basically moving almost throughout the entire enclosure. Well, from these maps, we can see that this guy is intruding onto the territory of this male, as well as onto the territory of this male. And it turns out that when we did the paternity analysis, he actually sired young with a female mated to this guy, bonded to that guy. We can do this for another enclosure with another focal male. And here we have somebody who mated only with his mate. And you see he's not intruding on the territories of other males. And if we start to put those data together, we see something interesting. First of all, we see that that whether males engage in extra pair fertilization or extra pair paternity, we call those EPF males, if they do that, we find that they pretty much always lack vasopressin receptor in the retrospinal cortex. So this guy with low RSCV1AR is an EPF male. This guy with lots of vasopressin receptor is an IPF male, and those are their respective details there. If we look across EPF males, we see that they have very low levels of expression. If we correlate the levels of expression with their aspects of the space behavior, we find something interesting. First, this is something people often find confusing. First of all, IPF males are only residents. EPF males are both residents and wanderers. We don't see any difference between residents and wanderers in the retrospinal cortex. So it doesn't seem to be critical for pair bonding. What we see overall is a main effect in which for both, as the RSCV1AR gets higher, the rate at which they intrude on territories of neighbors goes down, and the rate at which they get intruded upon goes down. But that effect is more clearly stronger for the single animals. And I don't know if that's because of the difference in the scale of their home range and because we can only measure it as a function of home range size or not. But there is a significant main effect here. So when we kind of put all this together, what we think is going on, and there's some speculation here because there's some psychological details that we do not understand yet. What we think is going on is the retrospinal cortex is really important for memory and perhaps spatial memory in particular. And we know that the vasopressin receptor is really important for kind of signaling social context. And so what we think might be going on is that maybe these guys with the high levels of vasopressin receptor are really good, like they recruit this memory circuit when they're in social encounters. And so presumably that means they're really good at encoding the interaction between social stimuli and their distribution in space. And the way this could turn into the pattern of data we see, and again, this is a hypothesis, is that these guys may be very good at remembering territories they've intruded upon and been chased out of, and thereby be less likely to return to those. But they may also be good at guarding their own territories. And so what we think is going on is that this is some adaptive variation in which these guys are gaining fitness by being philanderers, but they're giving up fitness in the form of interpair fertilizations. And we actually know this is true. I'll show you those data here in a moment. Whereas these guys are really focused on effective mate guarding, and are, as a result, getting their fitness from a different place. So if we want to summarize that graphically, it looks something like this. If you're this guy, then what you want is to have high vasopressin receptor that's going to promote your guarding efficiency. But if you're this guy and you're only going to gain fitness by not remembering those places you've been chased off from, then it may actually be adaptive to have a different kind of cognition, one in which you have a poorer sociospatial memory that favors a kind of scramble competition. Well, this was our idea. And so what we've been doing is testing this in the lab with a variety of spatial tasks and different kinds of memory tasks. And we've had some trouble getting the control memory tasks to work as robustly as we would have liked in some cases. But we did some ibotenic acid lesions of the retrosplenial cortex in these guys, and this isn't published yet. But what we found was that if you lesion the retrosplenial cortex, you don't actually impair their learning of a classic spatial memory task like the Barnes maze. That surprised us. That when we look more closely at the literature, it shouldn't have been quite so surprising. What surprised us even more, though, is that when we lesioned the retrosplenial cortex, we actually abolished the memory, or we actually abolished the partner preference. And so this is somebody given a PBS vehicle, and this is the time they spent with a stranger versus the time they spent with their partner when given a choice. These are highly different. This is an ibotenic acid lesion in which we selectively lesion the retrosplenial cortex. And what you see is they stop caring, that they treat the two as equivalent. We've since replicated this with dreads, in which we take a virus and put in a protein that we can use to selectively inhibit the activity of neurons. We put that into the retrosplenial cortex. And then in one group, we test them under the presence of a drug that suppresses the activity of those neurons. In another group, still has the virus, but they get tested without the drug. And again, what you see is the activity of the retrosplenial cortex seems to be necessary for the expression of this preference. So we're rethinking our field data and thinking maybe it doesn't have to do so much with space use as was our initial focus, or spatial memory. Maybe it's more about the memory for the identity of the mate. And so that actually fits with some more recent work on the retrosplenial cortex that suggests it might be a kind of long-term storage or an access point for long-term memory that then shunts that information into current ongoing experiences and allows it to be used. And so what we're doing now is some work on kind of engram related things. And so the idea here is to ask whether the retrosplenial cortex is actually storing a memory of your mate. And the way people are doing this in the comparative psych literature with mice is often to use to visualize the messenger RNA of a transcript that's associated with neural activity. And if you visualize that transcript 5 minutes after the stimulus that evokes the neural activity, that transcript is all near the chromosomes inside the nucleus. It shows up as two little focal dots like that. If you wait a while, though, that transcript gets exported out of the nucleus and into the cytosol, and it looks something more like this. Well, if you do something like, so what we're doing now is if you present a mate, say 30 minutes ago, wait 20 minutes, present a non-mate, a stranger, or do them vice versa, you can ask, to what extent does the nuclear label overlap with the cytoplasmic label? And the extent to which it does suggests a common representation of that stimulus, in this case of the mate. And so what we're predicting is that there's actually a representation of the mate that's present in the retrosplenial cortex, and that these individual differences in vasopressin receptor might be changing the size or the acuity of that representation. So that's kind of our current hypothesis and what we're doing with that. Unfortunately, I don't have those data yet, but that's work underway. So now we're going to go down a level and ask about genetic variation and how it gets translated into this phenotypic variation. Before I do that, let me pause for a second and see if folks have questions.
Must be crystal clear.
It's about to be not so clear. Yes, in the back.
So please go to the mic.
I can repeat the question if that would be.
We talked a little bit about maybe some scalable applications.
Are there scalable applications in humans that this research has? So, I mean, I think that that the work I've talked about so far, like, I don't think that that unfaithful men have poor vasopressin receptor in the retrosplenial cortex. Like, that's not the motivation for what we're doing. It's not the interest. I think what is interesting, what is motivating, and this will be more clear as we go into the genetic work, though, is that I think the application is, I mean, it's not an application in the sense it's not solving a problem. It's a conceptual application. I think the point is, what's natural variation look like, and how can we identify adaptive variation? And I think that's a really kind of big, interesting question. And even opening our ideas to the possibility that there might be adaptive individual differences in complex cognition, something as complex as long-term memory, I think is kind of a fascinating idea. And it's this sort of idea that wouldn't come naturally from within psychology or traditional behavioral neuroscience. Yeah. Okay. So I'm going to tell you a little bit about the vasopressin receptor gene, which fortunately is a relatively simple gene. It is the vasopressin receptor gene is called AVPR1A, and it has just two protein coding regions. So there are two exons, exon 1 and exon 2. And then here in bright yellow corresponds to the part that actually codes a protein, and then just upstream of that are parts of the transcript that don't encode protein. So there's just one intron. That's convenient. And then there are some repetitive sequences in and around the gene. And so this microsatellite here has been studied quite a bit. It's pretty famous. So there's another microsatellite a little bit upstream of that, and another one in the intron. If we look at this length of sequence, so overall, this is about, the length that's represented here is about 8,000 base pairs or so. It's enough to kind of give you the immediate vicinity of the gene, but it isn't looking certainly exhaustively the possible regulatory sequences. If we go out and we sequence a whole bunch of individuals just caught from the wild or from our lab colony, we see there are lots of variable sites in this gene. And what we can do is, for each of those variable sites, maybe that variation will be a T versus a C, say. Well, for each of those variable sites, each individual will have a genotype. They'll be either TT, TC, or CC, right? And then we can ask, what is the, and we can code that as 0, 1, 2. So we can just run a little ANOVA and ask, how much variation in the abundance of V1AR protein can be explained by the genotype, right? Really simple. Well, that's basically the logic of a genome-wide association test, or in this case, a gene-wide association test. And that's what we did first. We had, for a variety of reasons, we thought this variation in RSC vasopressin receptor had at least partly a heritable origin, but we weren't certain, and then we certainly didn't demonstrate that with the data I've shown you. So the first thing we did was to look at all these sites that were variable at the locus and to find which of those, if any, were significantly associated with RSC expression. So here's the strength of association on the y-axis expressed as a negative log of p. So this is 10 to the negative 6th up here, and then down here is a traditional univariate p-value of 0.05. And then here is the position along the locus. And so each of these bars is a different variant that had a differing degree of association. So if we applied this very lax standard without any correction for all the different tests we did, we find about 30 or so sites that were associated with the expression. If we make that standard more stringent, we really find that after correcting for multiple comparisons, there are really just 4 sites. And interestingly, those 4 sites are all really tightly linked to each other, significantly so. And so really, this is segregating as just 2 alternative alleles, a high-expressing allele and a low-expressing allele. And interestingly, if you control for the genotype here, for example, all of these other positives disappear. So they're only showing up as positives through their linkage to this site. And so it looks like we've got actually something pretty simple, a high allele and a low allele. Well, how do we know if those sites are doing anything? We don't from this data. It's possible that they're simply linked to something else that actually is doing it that we just haven't included in our study. Well, to make sense of what might be serving a function in these regulatory regions and these non-coding regions, it's useful to consider just the scale of the genome and the epigenome. And so there are about 3 billion nucleotides in just 1/2 of the genome that you then have duplicated. Each of these nucleotides is 0.34 nanometers, something like that. And when you put those together, that comes out to about 2 meters of DNA per cell. You've got 30, 40 trillion cells in your body. It's enough to go to the moon and back over 200 times. So when you've got that much thread packed into one tiny cell, you have to think a lot about its packaging. Really what you need are a lot of spools. And each of those spools then can have information that designates what that piece of DNA might be doing. And so within the genome, those spools are nucleosomes. And those nucleosomes are made up of histones, and they can be packed very, very tightly, as shown here below, or they can be more open and ready for access if you need them, as shown up here. Well, if you look at these up here, what you see are marks on the epigenome that the cell uses to figure out where regulatory sequences are. And so there are really specific marks on the histones that indicate that something is regulatory. And so what we did was we went into I'm just realizing I don't have a slide on this, sadly. I usually do. We went into the RSC of the prairie voles, and we precipitated out the DNA that was associated with this mark for regulatory DNA. So this is a mark that's specifically for so-called enhancer sequences. And when you precipitate it out and then sequence it, you overlay those sequences that you get with your reference. And they end up being like little sort of peaks where the regulatory sequences are. And when we do that, what we find is that these 2 peaks here correspond to a strong signal of an enhancer within the intron of the vasopressin receptor. This peak here does not marked as an enhancer, but it's marked as like this constitutively active piece of chromatin that's associated with a tethering transcription factor. So the point is that these 3 things that are in almost perfect linkage with each other also reside within regulatory sequences, suggesting they are probably actually causal in some way. Okay. So that suggests that maybe it's worth going after these pieces in a little more detail. So the next thing I want to do is ask whether these differences between high and low expressing alleles really reflect the epigenetic regulation of the locus, whether they reflect aspects of transcriptional regulation. So what we did is we found these from kind of a broad screen. And so the first thing we did was to really replicate our our screen. We sequenced animals and figured out who was had high alleles and low alleles based on our definition. And then we made parents in which one parent had one high and one low, and so did the other. And the beauty of that is when you cross them, you get offspring that have 2 copies of the high or have 2 copies of the low, but they're siblings and they were raised by the same parents in the same nest. And when we do that, we can look at a lot of detail. First, we replicate the association with the RSC-V1AR. We don't see an association with any other brain regions that seems to be specific. If we look at the underlying vasopressin message, we see we can detect it in the transcript level. So it's not a change in the function of the protein. It really is a change in the transcript. And then we even see that individual differences in the transcripts are related to individual differences in the amount of receptor that actually shows up in this brain region. Okay, so we look a little more closely at this locus, and what we see then is that if you look at these sites, one of these 3 sites that's associated with expression is actually a polymorphism in something called a CPG site. Does anyone know what a CPG site is? No. I'm not sure why you say the P, but in DNA, when you have a C followed by a G, there's a phosphate in between them. Remember, DNA has 4 letters, right? A, T, C, and G. If a C is followed by a G, we call that a CPG. And the reason that's significant is that that C is a classic site that gets targeted during development for methylation. Methylation that can have an enduring impact on the expression of that gene. So what we have then is a polymorphism, a genetic polymorphism in the potential to be turned on or off by development. That's kind of interesting, right? And that's the sort of question that we couldn't have asked if we weren't looking at genetic variation per se. So it turns out that if you look at this high allele, that overall within this intron enhancer, this regulatory element, there are 4 CPGs. They're naturally there. And then there are 3 polymorphisms, none of which are CPGs in the high allele. But if we look at the low allele, they can all be CPGs. You can have 7 CPG sites in the same little segment that normally only has 4. And that suggests maybe a difference in the sensitivity of the allele to silencing. And so we started to look at that and actually look at the development of this brain region. And when we did that, we saw something that was really pretty interesting. This is the ontogeny of the brain variation in the first 2 weeks of life of a prairie vole. And so on the day of birth, there's no retrosplenial cortex expression, and we see that here. As they develop in these first 2 weeks, we see an increase in expression, but right away, as soon as we detect expression at 1 week, we already see these genotype differences. So clearly there are differences in the very unfolding of this brain pattern, which then we speculate are going to have lasting consequences on their social cognition. If we look at the methylation state of the alleles, even just focusing on those that are not variable, we see a difference where the high allele has less methylation than the low allele. So overall, this kind of paints a picture in which we've got genetic variation that then influences the sensitivity to developmental environments. We've done some developmental manipulations, and the alleles are differentially sensitive. Then, then this environmental variation, genetic variation interact, shaping the adult phenotype, and then that adult phenotype may be contributing then to their sexual fidelity and space use and more complex memory and cognition in the wild. Okay, so this brings me to my last part. Has selection on retrosplenial cortex vasopressin receptor maintained variation at the AVPR1A locus? So now this is really asking, we've seen that there's this variation there. We've seen that it predicts important aspects of behavior. Can we actually detect a history of selection at the locus? And we're going to do that in 2 different ways. One is a kind of a more traditional behavioral ecology kind of approach, and one is a more evolutionary genetics approach. So if we just look at the fitness of the 2 alleles, now we have these markers, and we go back and genotype all our animals, and we say, like, what's the relative fitness of the high allele and the low allele? And they're basically the same. There's no difference. But if we go back and say, what's the fitness of each allele in the context of intrapair fertilizations only? Or what's the fitness in the context of extra-pair fertilizations only? When we do that, we see that there's a real difference. Basically, the low allele is the only way that animals are engaging extra-pair fertilizations in our study was if they had the low allele. In contrast, and so the low allele is getting much of its fitness from extra-pair fertilizations. The high allele is getting its fitness entirely through intra-pair fertilizations. But on balance, these two things balance each other out. Okay, so that's nice. But any behavioral ecologist who's done tests of selection in the field would probably be a little uneasy about those tests because they're just a snapshot in time, right? But fortunately, because we've got genetic data, it's not the only thing that we need to look at. We can look more deeply. And so we can test for something called balancing selection to ask if there's a history of selection on the locus that's consistent with what we think is going on. And so this is how it works. Imagine you were to take a piece of DNA and you were to look at it. And so this is kind of the green is sort of a reference sequence. And then here in orange is a departure from that reference sequence in some way, a sequence that's not what you expected to see, right? Evidence for a mutation. So there's some polymorphism at that site. Well, as you look through the different examples in a population, you start to accumulate different polymorphisms that are distributed on that locus. Well, we know from basic theory that when a new mutation happens, it's present on just one copy of the chromosome, right? There's only a single copy of the first mutation. And oftentimes that new mutation just by chance gets lost. And so most new mutations are very rare. And if we look at the distribution of frequencies of mutations in the genome, that's what we expect to see. And so if we did that in the prairie voles and we actually looked at either genome-wide or in this case, which we've done also, or in this case, just a few putatively neutral sites, what we see is that most mutations are at a frequency below 20%. And as the frequency goes up, the number of the mutations we find at that frequency goes down. And that's a very common pattern that we expect in the absence of selection. But what we think is going on is something different, right? What we think is going on is that we have some variation at the vasopressin receptor locus, but somewhere along the line, some new variant came along here that produced adaptive variation in behavior and then contributed to, and then was subject to selection and actively maintained in alternative forms. So what that looks like is a new variant emerges and then rises in frequency in the population, dragging the other stuff that was linked to it along with it. And now what we have then are a bunch of linked sites that are at intermediate frequencies we didn't expect to see. And that's a really unusual pattern, and that's diagnostic of something called balancing selection. So if we look at the vasopressin receptor, that's exactly what we see. What we see is a real excess of these high and intermediate level frequencies that shouldn't be there and that are not there in the background of the genome as a whole. So that tells us that selection actually actively has maintained variation at the locus. And if we look where that signal is strongest, it overlaps with the signal that we get on the RSC. Okay, so I'm going to summarize now. So we kind of talked about this as an evolutionary problem in which selection is potentially maintaining alternative kind of cognitive phenotypes. At least that's how we're thinking of it. Below that is a population level problem, an ecological problem of home range exclusivity. There's a psychological dimension in which we think there's either sociospatial memory or just social memory per se. Beneath that, we have neurobiological variation, epigenetic variation, genetic variation, and then all those sort of feeding back on each other. And so selection operating at the level of a gross behavioral phenotype, then we can imagine now how that could percolate all the way down into the details, molecular details of the genome. Okay. So does the vasopressin receptor explain why some people are more faithful than others? I don't think so. But there is this interesting study that looked at a specific length of a microsatellite um, and asked people whether they experienced the threat of a marital crisis or threat of divorce in the last year. If you had 0 alleles, then the likelihood that you answered yes, if you had 0 of this particular allele, the likelihood that you answered yes is about 15%. The same if you had 1 copy. But if you had 2, this likelihood went way up. Now, overall, though, I think, so this is kind of suggestive, it's interesting, it's been cited a lot, but the first author of this paper thinks that this may have just been asking too many questions and is not very confident that this result is actually a real one. Overall, I don't think that what we're doing is trying to explain human fidelity. We're not trying to explain why some people are faithful and some are not. We're not making an argument that there's a genetic basis for a variation we see among people. And so I would say that if you wanted to, get your bonding gene test and find out about the microsatellite length of your vasopressin receptor, it might be $99 badly spent. A better thing to do would be to have a conversation with your partner. And but really, that's not the point of the work, right? I mean, really, the point of the work is to think about individual differences and to frame them not in the context of health and disease, but to frame them in the context of natural variation and function. So, so then our conclusions are that love and attachment seem to depend on reward and memory in an interesting way, much as Garcia Marquez suggested, that selection is shaped genetic and epigenetic variation in the social brain, at least in the case of the prairie vole in this one measure. We suspect that variation in the social brain, including genetic variation, may actually be commonplace. And really, I would like to emphasize that neuroscience needs to think about genetic variation as biology worth explaining and worth understanding rather than as noise. So I'm going to conclude with kind of a big picture here. This is a quote that I'm really quite fond of. It's a quote by Henry James, coming back to him once more. He says, "The self-same atoms which chaotically disperse made the nebula, now jammed and temporarily caught in peculiar positions, form our brains. And the evolution of the brains, if understood, would simply be an account of how the atoms came to be so caught and jammed." So I love this quote for a variety of reasons. It's kind of lyrical and beautiful, I think. It's also entirely facetious because when he says this in Principles of Psychology, he's really making an argument against material explanations of the brain. And he goes on from here to talk about these molecules as being a kind of mind dust or fairy dust, that there's something about the brain that's irreducible. And as a contemporary neuroscientist and molecular biologist, my view is different. My view is very materialist, and but to me, there's something really beautiful about the fact that something that seemed, in his day implausibly fantastic, now is really fantastically plausible. This is something we can study with the tools of neuroscience, of evolutionary genetics, of molecular biology, and get some new insights into why brains vary and how they vary. Of course, this wouldn't be possible without some extraordinary people in the lab. Miriam Okavat was a graduate student who finished her PhD and is the first author on this really lovely work on the association study. Alejandro Barrio was a wonderful molecular biologist. He's now at Duke, and he did the evolutionary genetics work I talked about. Gerard Wallace did some of the field work as well as some of the association studies on the brain side. Alex Ophir, you guys have met. He was just a wonderful human being, and I was lucky to have him as a postdoc for a while. Same is true of Lauren O'Connell, who helped us set up the ChIP-seq stuff. She's now at Stanford. Zara Dagani helped us do the initial association work. And then there are a variety of other folks in the lab that are really critical for our ongoing work. And then the National Science Foundation, who funds all of our work so far. So with that, I will gladly take questions with the time that remains. Thank you.
Of course, the theme of the summer school is not sexual bonding, but there's a reason why we invited your group over. And so I'll ask the more general questions. I mean, pair bonds are particular affiliative behaviors. Can you say anything about to what extent this generalizes to questions about other sorts of affiliation? Parental, that's related to pair bonding, familial, extrafamilial, species, even species-based.
Yeah, I mean, I think of bonding. If you think about bonding in the psychological sense with its tradition of attachment theory and those things, you think about it as a behavioral ecology phenomenon. I think those aren't really so far apart as far as I'm concerned, in that bonding is kind of fundamentally about throwing your lot in with somebody else, with deciding that their fitness and your fitness are tied. And that's how I think about it as a behavioral ecologist. And in humans, those bonds take lots of forms. They take the form of parent-infant bonds. They take the form of romantic bonds. They take the form of social bonds. If we look across the animal kingdom, we see each of those kinds of bonds as well. And I presume that those bonds all have common cognitive needs, right? They all require memory for an individual. They require, the that sort of common sense that your outcomes are tied to each other. I think those are. I think those are likely to involve at least an extremely overlapping set of processes and mechanisms because they have similar demands.
If you'd asked me that question that you asked at the very beginning, I would have said that there is something similar, not just addiction, but something similar with imprinting. You have a feeling that you've sort of imprinted on one person. Is it actually related to imprinting?
That is a wonderful question, and I think that's something that we don't know. When I think about the work of imprinting, I'm thinking, for example, of some of work on olfaction and gustatory responses by Robin Hudson at UNAM and Gabby Nevitt, I believe, at Davis. And in both of those cases, Nevitt did work on salmon imprinting on their nasal streams, and Hudson did stuff on infant rabbits imprinting on food that their mother ate. In both those cases, what you have is kind of like a critical period in which in which the sensory system has been rewired to assign a particular valence to some aspect of the world. Now, I think that the extent to which bonding is something you only get to do once, it makes sense that it might do something similar. And in fact, when prairie voles bond, at least in the lab, females have an induced estrus, and so when females in a lab situation, they're generally they've never mated before. They haven't even necessarily gone through puberty, and the induction of that puberty is part of the process of initiating the bonding between the male and female. With that induction of puberty, there's a huge increase in the number of new neurons that are going out of this out in this rostral migratory stream into the olfactory bulb, presumably also into the hippocampus. And I think it's totally plausible that the effect of that is to give particular stimuli, access to particular valence. In which case, it would be very much like imprinting developmentally. But really, that's not been looked at, and that's my speculation. That's kind of something I've long found interesting, but it's not something that I've ever seen folks work on. They've described this olfactory, these new neurons, the neurogenesis associated with olfaction, and the onset of bonding. But exactly figuring out what's going on there, that's not been done.
Okay, now we've got Steve here. Don't lose your opportunity to ask what you're going to probably ask offline anyway. It would be very disappointing if, of all people, Steve Phelps didn't inspire the students to ask the kinds of questions that they've been asking in every other session. I'll fill up the time with a personal one. I conjectured to to Larry that the reason my romantic life was ruined was because I have an excess of vasopressin receptors. Does
and that ruins your romantic life.
I'm because
really
it
curious
produces
now.
because it's not just fidelity. I don't know about
Yeah.
that, but it's also territoriality on your mate. And
Yeah.
that means it produces toxic jealousy as well.
So an excess of mate guarding behavior. Yeah, it sounds like you might have very high retrosplenial V1AR.
You just said we're going to talk about males today. It's just because that's what you study the most. But what about females in that?
Yeah.
Do they, by their own choice, go extramarital, or do they jump to
Yeah.
I don't know how you say that in English. But
one interesting thing about the math of extra-pair fertilization is that exactly as many females, like the average degree of fidelity between sexes has to be exactly the same, right? Because every time a male engages in extra-pair fertilization, so does a female. So the females definitely do it. They show if you look at the home range size as a function of bonded versus not bonded, in some data sets, we get a sex by bonding interaction in which there's a change in both sexes, but the change is greater in males. And the way I think of it is analogous to the old framing in the behavioral ecology literature that females maybe are following resources and males are following females. And so I think that the females are definitely showing individual differences. They're showing individual differences in sexual fidelity. They're showing individual differences in space use. But those differences, to me, at least in the space use data, don't seem to be as profound as the male differences. And they don't seem to be as reliably aggressive in response to bonding either. And so my interpretation is that from an evolutionary standpoint, they might be less invested in the bond itself. I think the bonding for them may be more about infanticide deterrence or something like that. But that's speculation. Thanks.
Okay, to offer you a challenge for humans at least, how does culture push this around?
Yeah. So how does culture push this around? So where to begin on a question like that? It's fascinating. I don't know where to begin with respect to the biology of our mating system as humans, right? Because if you look globally, there's a lot of variety in the mating systems that we have. If you look at things that are good markers in other primates, like relative testis size or things like that, those things are sort of complicated when you think about people. They really only speak to sperm competition. The genetic data suggests that males have a smaller population size than females, suggesting some slight polygyny. So I guess my point is I think that our mating is very fundamentally fluid, and it's hard to disentangle cultural and non-cultural sources. I mean, clearly we bond, but whether those bonds are naturally exclusive, I think, is such a value-laden question. I don't feel like I could answer that as a scientist conclusively. I think I would have my own ideas, but I don't know. Yeah. I'm not opposed to sort of evolutionary explanations of human behavior in the sense that obviously humans have been subjects of selection just as any other species has. But I am opposed to sort of simple deterministic explanations that don't recognize the obvious complexity of our behavior or treat it as something in which we have no choice. But, yeah.
So earlier on, you described an experiment where you have those those voles inside a perimeter, which is very similar to your colleague Ophir's, Mr. Ophir's presentation. We heard about some of that. And so you have those more stable couples, right? The males that are territorials, I think you called them, and the wanderers. And I'm wondering if the behavior of the wanderers is in any way associated to the population of females. So, for example, if you put more females, would some of them remain wanderers for life, or would they, since they have more choice? So, do you manipulate the number of females in order to get those results, or do the wanderers really are hypothesized to? That's how they go.
Yeah. So certainly, I mean, when we look at our data, it looks like the animals that are wanderers are wanderers because they can't monopolize a female, and that they would switch to becoming a resident when they're able to monopolize a female. So I don't think that they're stuck as wanderers. I think they're waiting for a partner. But we don't Can you remind me of your question? I've lost the thread.
Yeah, I'm just wondering if not wandering, but wondering if sorry, couldn't help.
That's again, no judgment.
Came up. Okay, so I'm just wondering if the number of females has an impact. Limited number of females has an impact. Or if you put more, like unlimited females, the wanderers would all become territorial.
Yeah, so if there were a limited number of females, certainly the wanderers would form bonds and would become territorial, certainly. And Alex has actually played with sex ratio in the field a bit. We're starting to get some higher resolution data, both from like tracking more and also from some automated telemetry that we're doing. And I occasionally see what looks like a male with two females, for example. And I think that whether a male might, or female might be able to bond with more than one animal is something that, they're usually really aggressive. It's certainly in the lab in a way that precludes that, but we occasionally see hints in nature that's a possibility. And so I would think that if you had somebody who had been a wanderer and the sex ratio was heavily biased, that certainly he would mate and form a bond, but he actually might try to monopolize more than one female.
All right, thank you.
Yeah.
Hi, I'm not sure if this has already been mentioned, but I was wondering about what happens when the bonded pairs are separated. Like, do they go through some sort of grieving period, or?
Yeah, that's a great question. A friend of mine, Hemu Nair, and Oliver Bosch worked on that a bit in Larry's lab as postdocs. And Oliver, I think, has maybe continued to do some of that work. And yeah, they do show things that look like grieving. And so, like in classic depression-related tests, like forced swim tests, for example, like they give up swimming faster, is my recollection. When they've been separated from their partner. I think, if we're drawing the analogy between the bonding of these guys and human bonding, there are a lot of fascinating questions to ask, like, can they bond a second time after they've been separated by a certain amount of time? But we don't, surprisingly, that's a simple study that nobody has done to my knowledge. And to what extent does sexual behavior offset the cost of that emotional behavior? Like, is there something like rebound sex in prairie voles? I wouldn't be surprised if there was. I think that's plausible. I think as emotional cognitive mechanism is probably something pretty general, right? So anyway, that's a fascinating question, and it opens up a whole lot of unstudied things that I think would be fruitful.
Thanks for the talk. This is a little bit of a silly question related to physiology of vasopressin. I was making an analogy in my head to oxytocin, which is also being related to bonding. But the peripheral effect of oxytocin in the body, I think you can also relate it to some things in bonding, like, for example, lactation in females or like contractions of the uterus. But it seems to me like a little bit, when I think of vasopressin as like the antidiuretic hormone that helps you like get water back to have like control blood pressure. And then this same molecule in the brain is also related to bonding. Do you think there's any like biological or
Yeah.
evolutive
Yeah,
relationship between these two functions, or is it just a coincidence?
I mean, we certainly treat them as though they were completely unrelated, and it makes it easier for us to talk about them. But at some point in the distant past, there was something it was doing, and the term vasopressin comes from its role on the contraction of smooth muscle and arteries, right? And so it's possible that that vasoconstriction, I can imagine, blood pressure is something that's kind of fundamental to arousal, and maybe that's an ancestral function that predates both of these. I don't know, honestly. I think that's a great question. It's something that comes up from time to time, and it's something that none of us really understand. I think to understand it, you'd have to take a deeply phylogenetic comparative approach to ask about to see what you could infer about the origins of these functions. But that's a good question.
I just have a follow-up question on the previous question about culture. You sort of took it to mean human culture alone. And if I understood you correctly, you said that you're not comfortable reducing human behavior to evolutionary explanations alone because of complex sociality and individual choice. And I was just wondering about your justification for not having the same view on culture, complex sociality, and individual choice in non-human animals, but reducing the behaviors to genetic explanations.
Yeah, it's not so much that I think behavior in humans is beyond the influence of genetics. It's not that I think it's immune to selection or evolutionary forces. It's that the kind of fluidity and ability to adjust behavior in humans is really profound. And so I guess the plasticity, I imagine, is greater and great across a range of domains. So that isn't to say that I think animal interactions, social interactions are necessarily simple ones. I mean, obviously we're hypothesizing now that what we're seeing are reflections of a long-term memory for specific individuals. I don't think that's going to be unique to the prairie voles. I think if that's true, that'll be something that characterizes bonding across, really a wide range of species. But I guess I think in humans there are more degrees of freedom in the expression of our behavior. And I don't know, maybe it's harder to interpret. Really what I was reacting to was it wasn't the idea that humans are special. It was really the idea that a really reductionist perspective on human behavior is often kind of too simple, or just not doesn't seem intellectually honest to me somehow.
But does for voles?
Yeah, I guess so. No, I don't think that. I mean, I don't know what the inner life of a vole is, and I had over beer. I had a really lovely conversation with Mike Ryan, who you guys also saw here. He's my colleague. He's also my advisor. Larry Young was my postdoc advisor. Alex Ophir was my postdoc. So we're all sort of related in a lot of ways. Anyway, Mike and I were having a conversation about what it would mean to demonstrate empathy. And we were talking about Larry's data and James Burkett's data on the behavior of the voles, like in response to their mates getting shocked, right? So I assume Larry talked about that work, did he? Yes, no? Yeah. Yeah, the comforting behavior. Yeah, so it shows a lot of things that look like empathy in that there's a behavior that's directed at the mate in response to the mate showing apparently some signs of distress. The giving of that behavior reduces the stress both on the mate and and on the focal individual. All kinds of things like that, right? It's really fascinating. And so Mike and I were talking about what data would convince you that they were showing empathy. And I posed a kind of thought experiment and said, okay, we have these tools now that allow you to image the active neurons using conventional IEG markers or whatever, but that allow you to do that in every cell in the brain, right? And so you can get a really complete map of what brain regions are active. So if you were to imagine that you were to do something like this experiment I just showed you with the engram, where you look at the mate versus non-mate, if you were to do that with another individual versus an experience that you had yourself, and you saw that those representations across the brain overlapped, like at what point would you call that overlap empathy, or what would you consider to be? So I guess my point is that I think that there can be these complex aspects of behavior in voles, but I don't know what their inner lives are is still opaque, right? And maybe necessarily so. And it's not clear to me at what point I would make that inference about their inner lives. But I think it's an interesting question. But yeah, my opinion would be that their inner lives are much less complex than ours, even though they have much the same machinery that governs those inner lives.
Thanks.
Yeah.
Yes, hi. Thanks for your talk. I guess I'm going to be asking a similar question I've asked one of your co-author earlier last week.
Okay.
So in preparation for your talk, I've read a bunch of your papers, and I'll be a bit more specific with your co-author. In your paper on methylation in the cortex of wild prairie voles,
You read that paper? Wow, I think you're probably one of the only ones. I'm delighted. It's a great paper.
Yes. Well, I just got stuck on one sentence in your methods section.
How many people would like to have a question begin that way? There's one sentence in your methods section on this arcane paper that I had a question about.
I just like further details on one quote when you say brains were frozen immediately on dry ice. Like,
yeah,
there's no more details. Like, how does that go?
Yeah,
it goes on.
So we took the brains and then we froze them immediately on dry ice.
Yes.
No. So yeah. So right. So there's no. I mean, the animals are anesthetized, and then after they're anesthetized, then
how might they be anesthetized? Just
we would generally use isoflurane anesthesia because it's fast and puts them out pretty quickly, and then you can assess whether the anesthesia is effective using standard toe pinch, tail pinch methods. And so when we take tissues, that's the way we would do it. And most of the things that we're interested in, the things that people doing mechanisms worry about are whether the onset of the anesthesia changes the activity of the tissues you're measuring. And so if you're measuring something like stress steroids in the blood, that it's really important that you do that you do the whole process within a couple of minutes. But in this case, we don't expect those changes to happen. And so it's not like we're comfortable in this.
So the process of the euthanasia doesn't have any impact on the results.
Yeah. And I mean, that's certainly the assumption. And so we're comfortable using anesthesia within the context of the questions we're asking. And certainly, we would have to have a really compelling reason not to, scientifically. And that's not the case with this.
Great, thank you.
Yeah.
That sounds like we're at the end of 10 days, and I think there's a little bit of fatigue setting in as well. But get your energies back up again, because at 4:00 we start again. Is there another? Oh, you have another one.
Yeah, thank you very much. I'll probably have to watch it again the talk to understand it completely. But I wonder, do male voles behave only aggressively with each other, or would you see sometimes within wanderers more bonding or friendliness or cooperation within with other males?
In general, the prairie voles, the males are extremely aggressive with each other once they've bonded. Before they've bonded, they're not aggressive. They do have an overwintering behavior in which if they're on short days, in the wild, the short days would correspond to cold weather in which they might overwinter and be more permissive. I think if I remember the natural history correctly, in prairie voles, those overwintering groups are generally family groups, so they still wouldn't be anything like bonded to each other. But Anneliese Beery has been studying the short day bonding between meadow-voles, which corresponds to overwintering groups. And there you might see something analogous to bonding. It wouldn't be like a pair bond exactly. It'd be more of a social attraction and social tolerance that was appropriate to the ecology.
And how would that relate to vasopressin or their levels or the expressions of the genes and stuff?
Yeah, that's a great question. Yeah, I haven't followed what Anneliese has done with that recently, so I couldn't say. I don't think. Yeah, no, there are things I could imagine. Like I could imagine that there might be differences in this sexually dimorphic vasopressin region that I mentioned, because in some studies it's been testosterone sensitive. And so it's plausible that when you go through short days and your testes shrink and stop making testosterone for the winter, that there might be some change in the vasopressin system as well. What that would do, or if that's the case, I couldn't tell you off the top of my head.
And just one follow-up, last follow-up. Would the females be more sensitive to males with lots of testosterone, or would that have an impact?
Yeah. So when we actually looked in our field data at what predicted who formed bonds, what we found was that they didn't care that much about the size of the males, although the males were all pretty similar in terms of size and weight. But there was an association between the anogenital distance of males and whether they formed bonds. And it's a little hard to tease apart the causal arrow there, because it could be that their testes got larger and there was a change in their AGD in response to bonding. But in the lab, we showed that females prefer males with a larger AGD. Overall, we know that males would prefer to pair, and they have a higher fitness if they're paired than if they're single. And what we think is going on is that, like, I think when males form a bond, they have to mate multiply over an extended period and release vasopressin for a long time in order to make a bond. I think the reason for those mechanisms is they're assessing whether or not they can effectively monopolize a mate before they form a bond with that mate. And so I think in the field, when they don't form bonds, is because they can't monopolize a mate. And along with that is that females seem to prefer these large AGD guys who presumably are more aggressive and more able to defend the territory.
Thank you.
Is there any relation between aggressivity and the status of the female? Estrus, pregnant, neither?
I don't know. I haven't seen that looked at, but that's certainly something one would predict would make a difference. And Mike Firkin did some lovely work in meadow-voles suggesting that they can have an exquisite memory for interactions that are associated with the timing of ovulation in meadow-voles. And so I think there's interesting work to be done there, but we don't know.
Okay, with that, I'd like us all to thank you very much for your talk.
All right, thank you.